Good morning, good afternoon, good evening, together with Brian Fox from the University of Michigan, and on behalf of the FUS Foundation and on the IUM, and on behalf of the organizing committee, it's our pleasure to welcome you to this fourth session of our workshop series on detecting, mapping, quantifying bubble activity in therapeutic ultrasound. So we already had 3 sessions, the first one on cavitation detection equipment, including hardware, software, the second one on cavitation localization and mapping, and the third one last week where we discussed cavitation threshold and doses. These sessions have been recorded. The recordings are available on the workshop website, together with a written summary of the highlights of the discussions. Today's session is about cavitation agents, what agents, for what applications, what characteristics. We will follow the same format as for the previous sessions. So first part will be presentations followed by 5 minutes of Q&A. We'll take 10-minute breaks and we'll resume for a panel discussion with the presenters and additional panelists. I just want to remind you that you can ask questions at any time using the Q&A section of the web page. The Q&A board will be reset after each presentation, but we are keeping track of all the posts. We really encourage you to post any question and comments during the presentation so that they are available for the 5 minutes of Q&A time. And this Q&A panel, of course, will be active during the panel discussion for you to participate. So now I'd like to introduce and to thank Professor Tyrone Porter from the University of Texas and Professor Brian Fox from the University of Michigan for organizing and moderating today's sessions. And Professor Tyrone Porter will now introduce the session.
Yes, good morning, everyone. This is a really exciting workshop and really excited to be a part of this. We have, up to this point, as Fred has mentioned, we've had a number of sessions on monitoring cavitation, detecting cavitation, mapping, imaging, displaying cavitation, and dosimetry. And while ultrasound can be used to nucleate cavitation without any exogenous agents, it can be actually helpful to introduce cavitation with nucleation agents, nuclei. And so today we're going to have a session in the conversation, hear from experts, presentations, as well as panelists on the different types of cavitation agents that can be generated, manufactured, and then how does one actually characterize. And so today our first speaker to start us off is Kang-Ho Sung. He is a research associate at the University of Colorado in Boulder. He studies microbubble and focused ultrasound applications for drug delivery and neurotherapy. He is currently a senior postdoctoral fellow working with Mark Gordon at Colorado Boulder, and he is also a chief technology officer with Advanced Microbubble Laboratories. He received his PhD in mechanical engineering at Colorado Boulder, having received his master's in BME at Cornell and his undergraduate degree from Yale University. So he's going to start us off with a presentation on the pros and cons of microbubbles as a cavitation agent for therapeutic ultrasound.
Hi, everyone. My name is Kang. I'm a research associate at CU Boulder, and today I'm very privileged to have the opportunity to speak on some of the pros and cons of using microbubbles as cavitation agents for therapeutic ultrasound. So just to get started, the requisite description of what microbubbles are. This is a rather large lipid microbubble, but most are 1 to 10 microns in diameter with a hydrophobic gas core to resist dissolution, stabilizing shell, and this one has a PEG brush layer to reduce immunogenicity and help resist against coalescence. There are other shell materials that are suitable for microbubbles out there, and each one has its own unique mechanical and physiochemical properties. For example, sugar and protein microbubbles may be more rigid than lipid microbubbles, while lung surfactant microbubbles have very foldable or wrinkly shells that allow them to potentially resist shell material loss in extreme expansion and compression events. An expansion and compression of microbubbles can certainly occur in the low pressure and high pressure regimes of ultrasound waves. And as ultrasound waves pass through the microbubbles, they cavitate, and 2 broad families of cavitation are stable and transient cavitation. I think it would be fair to say that neither mode of cavitation is inherently more destructive, but that both are capable of achieving a wide range of bioeffects depending on the ultrasound and microbubble parameters. In practice, microbubbles are generally injected, and if they are injected to the bloodstream, they will circulate through the entire body. In blood-brain barrier disruption, microbubbles that are flowing through the brain will be targeted with ultrasound and used to transiently permeabilize the brain vasculature to enable drug delivery. In the case of tumor therapy, you could imagine that this technique could be used to enhance the permeability of tumors and enhance drug delivery, potentially allowing us to reduce the dose of chemotherapeutic necessary. And in the case of something like lithotripsy, microbubbles might be used to achieve a higher level of mechanical force on the kidney stones and improve the breaking of the kidney stones. So just to start, we'll look at microbubble pros. And it's good to look at another target efficient drug delivery method to put them into some context. So one method is intracranial injection, which is highly invasive and can cause quite a bit of lasting permanent damage to animals and the brain. This effect is evident even in their behavior several weeks after the procedure. Thankfully for microbubbles, the brain is highly vascularized. And an interesting fact is that most cells within the brain reside within 100 microns of the nearest capillary. Most cells within the body reside within 200 microns of the nearest capillary. So this suggests that maybe microbubbles in focused ultrasound could target a wide range of tissues and can be quite versatile. But to start on the official pros list here, the very first pro of MB-FUS is how efficient it can be. With microbubbles in focused ultrasound, even newcomers to this technique still manage to achieve the desired therapeutic result, even if the only thing they have is a rough set of initial parameters. So I think that's really unique to MB-FUS in the field of drug delivery. Number two is that the microbubbles add a new set of parameters that may be adjusted to either enhance a desirable bioeffect, such as kidney stone breakage in lithotripsy, or to reduce unwanted bioeffects, such as cell and tissue damage in something such as blood-brain barrier disruption. We'll speak more on that later. The third pro is that, as you saw, there are many different types of microbubble constructions and many are yet to be unexplored, but they all have implications for pharmacokinetics, immunogenicity, or whether you'd like to attach a targeting moiety or conjugate a payload to your microbubbles. The fourth pro is that MB-FUS can target very small regions of the brain. And additionally, it can target multiple regions of the brain, or if you broaden your focal zone for ultrasound, you could target a larger volume of the brain. The fifth, perhaps flagship advantage, is that MB-FUS is non-invasive. And the sixth pro is that its effects are generally transient. So in the case of brain vasculature compromise, given appropriate parameters, the vasculature will mostly heal within 24 to 72 hours. So the effect is transient. Seventh is MB-FUS is non-heating, which could make it more appropriate for temperature-sensitive regions of the body. The eighth pro is that MB-FUS is very cost-effective, especially compared to some of the contemporary cancer therapy methods, invasive transcranial methods, and of course, their post-operative costs. So those are the pros. And moving on to the cons, I'd like to handle this differently, just using a hypothetical situation in which a lab has a focused ultrasound unit already, and they're interested in using it for therapy. Their primary concern will be the ultrasound parameters, followed by how the tissue will affect the ultrasound signal. But with the addition of microbubbles, we have several new microbubble parameters, as well as physiological parameters that may affect microbubble behavior in the body. So this can pose a significant barrier to adoption. And the second con is related to those parameters, which is that if there is a parameter that is significant to your application and it is not controlled, this can lead to issues of unpredictability and inconsistency in the results. The third con is that microbubbles are inherently limited to the cavity or the vasculature into which they're injected. The fourth con is the added regulatory burden of adding this component, this new component of microbubbles, to your existing therapeutic protocols. This next slide is not meant to be read in detail, but these parameters correspond to some of the parameters that were adjusted in 3 studies showcased in the next slide. And so this parameter set is here to demonstrate some of the significance of controlling microbubble parameters. One of the very important outcomes that we and many people in the field look at is safety. So in the early days, a gross indicator of safety was red blood cell extravasation or microhemorrhaging. And even if these petechiae would clear after an MB-FUS procedure, after 7 days, the community was interested in some of the non-visual and less obvious indicators of safety, such as neuroinflammation. In this study, EGFP packaged in AAV was delivered with MB-FUS, and even 14 days after this, the MB-FUS procedure was conducted, there were signs of activated microglia or neuroinflammation in the target sites. Of course, in this study, it's difficult to ascertain whether this inflammation is from the viruses or the procedure itself. So many more very comprehensive studies were conducted by the rest of the field, and one of these was Dr. Kovacs, or Sophia Kovacs's experiment, which she isolated MB-FUS effects on neuroinflammation and showed that within 24 hours of the procedure, there was significant neuroinflammation. One controversial aspect to the study was the microbubble dose, or a microbubble parameter that was used, which only shows, goes to highlight the importance of controlling and understanding microbubble parameters and the significant challenge that that can frankly be, especially to the interaction of parameter sets between studies. The third result was from Dr. Hinnanen's group, McMahon et al., and this is a H&E stain of a rat brain slice, but with very reasonable MB-FUS parameters, or what many would have previously considered to be very reasonable. In panel B, McMahon et al. Demonstrated that neuronal damage could occur even with these parameters. And so what this just goes to show is how sensitive certain types of tissue may be to microbubble parameters and how important it is to control them. So we're going to tackle these cons by examining this waterfall, which is generating microbubbles in much the same way that the rest of us do, which is mechanical agitation. And mechanical agitation is excellent because it produces a very high yield of microbubbles, but fortunately, when gas is entrained in liquid, there can be many different volumes that are entrained and, of course, many different sizes. And this wide range of sizes is evident in commercial ultrasound contrast agents such as Definity, which are generated with mechanical agitation. So in the size distribution plot with size on the x-axis and volume fraction corresponding to size on the y-axis, you can see there is a wide range of sizes. And this variation can extend to the number of bubbles and can extend between batches. So different batches may have different numbers and sizes of microbubbles. Well, this is a problem because even size alone has significant effects on microbubble passive dissolution characteristics and how it responds under ultrasound. In this figure on the right, concentration matched 2, 4, and 6 micron diameter bubbles were placed on a benchtop and exposed to 0.5 megapascal, 1 megahertz ultrasound. And you can see that with 2 micron microbubbles, the much smaller microbubbles, there was a dramatic decrease in the number of microbubbles that could survive at these ultrasound settings. When we look at mechanical output of microbubbles, or more specifically in this case, predict the mechanical output, size also matters. So as size increases, the relative power output or the relative radial expansion of microbubbles at that size may decrease, while because of their persistence, power times time, the energy output of those microbubbles can increase. And this is indeed evident in vivo. So in blood-brain barrier disruption, where in this case, Evans blue was used as an extravasation marker, we see that 2 different sizes of microbubbles can achieve very different permeabilizations of the brain. And these are concentration matched as well. So this takes us back to those batch-to-batch variations regarding size and dose we were talking about. And regarding dose can be a bit of a pro and a con. It can complicate the picture and make it a little bit more simple at the same time. For example, in this case, there was a simplification using dose. So 2 different sizes of microbubbles, but injected at identical gas volume fractions of the actual microbubble gas, showed that they could achieve the exact same model drug extravasation in BBBD. But unfortunately, what we don't know is how these sizes might affect other bioeffects, such as neuroinflammation and microhemorrhaging. So these are all things to consider. And while more characterization may help us collapse more parameters and associate them with specific bioeffects, this is all challenges and cons to employing therapeutic microbubbles. Pharmacokinetics is also a pro and a con. While microbubbles are famously biocompatible, or at least believed to be very biocompatible by many people. Unfortunately, if you look at this figure where we sampled blood from the jugular vein after injecting various doses of microbubbles, you can see that the clearance of microbubbles from the blood is very rapid. And so this has implications for how and when you want to administer your ultrasound, as well as how you want to administer your microbubbles. The second point I want to make here is if you look at the green line, which was the highest dose, 40 microliters per kilogram of microbubbles, the pharmacokinetic response was not so predictable. So how do we solve these problems? Well, pharmacokinetics and pharmacokinetics predictability may be addressed with novel constructions of microbubbles that may either persist longer or be less immunogenic or be not as easily filtered from the blood and have physiological responses triggered. And we could potentially do that with something like this, which was a simple 2-component non-lipid formulation that we use to try to reduce immunogenicity and increase circulation persistence time. So in closing, I think that many of you are starting to realize that many cons are addressable with at least one of the pros that were also mentioned. And that was indeed the intention of this presentation. Perhaps I'm a bit biased because I work with microbubbles, but I think that is indeed the case. Many of the cons are addressable with pros. And to speak a little more on the added regulatory burden, I think it's best to put this in context of a procedure as simple as craniotomy, which the cost per quality adjusted life year, so this is how much it costs per year, is $550,000, over a magnitude greater than what is commonly accepted by clinicians and policymakers as an acceptable cost for medical procedures. So competing against something like that, I think microbubbles, focused ultrasound make a very good case for themselves. In closing, I'd like to quickly thank some of the people who were near and dear to my efforts, especially Angel and Peyton. Thank you guys for the very nice pharmacokinetics results. And I want to thank the ultrasound and microbubble communities. Your work attracted me to the field in 2010 and kept me in the field all this time. So thank you guys, be in good health and take care.
Okay, thank you so much for that presentation. I think it was a very, really cohesive and thorough discussion on some of the pros and the cons with regards to the microbubbles. There are a couple of questions that were posted. The first one here that I have is a question on APFEL on transient cavitation. So APFEL, Land, and Holland moved away from the designation transient cavitation some years ago. The bubble activity is not temporarily transient. There's some data that has been presented by Dr. Holland demonstrating sustained inertial cavitation from DEFINITY infused through an ECOS catheter. Why did you use this particular term in your talk?
Thank you, Dr. Porter. So the simple reason for that is simply because, you know, in the case of something like jetting, fragmentation, et cetera, the event is simply just transient. That was the only reason why. So.
Okay. The next question I have here is from Dr. Probst, actually. He's asking a question with regards to slides 10 and 11 in your presentation, where you show there's a dependence on some of these bioeffects with regards to the size of the microbubbles. So how dependent were these size dependence due to the ultrasound frequency used?
So that's a great question. And straight out of the gate, no other frequencies besides 1 megahertz were tested for those results. But it makes you wonder, you know, if you increase potentially not just the relative radial amplitude that these microbubbles are achieving, but if you increase potentially the velocity due to a higher frequency or slow it down, how could that affect the results? And it could be that higher frequency given equal radial amplitude could potentially destroy the microbubbles even more quickly or basically have a negative effect on their persistence. Of course, assuming that the microbubbles are oscillating to the same amplitude. as before. So just speculation, but definitely something we're really interested in.
We have a question from the audience as well, and I encourage audience members to please post your questions in the question and answer link, and we'll pose the questions to the presenters. So I have one question here looks like from the audience. Most or almost all commercial bubbles are polydispersed. So do you suggest monodispersed can be a better alternative? Very, very narrow size distributions. And how do you decide which size suits better for specific applications?
It's a really great question as well. So, I think there are many different ways to sort of approach the issue of microbubble parameters and some of the challenges. So one potential approach could be that we could collapse some of the parameters through the use of things like ultraharmonic monitoring to look at microbubble response. But another way is to potentially engineer in parameters like microbubble size so that we may not have to necessarily worry about, different sizes of microbubbles in a formulation responding in different ways. So I think that monodispersed microbubbles certainly have their place. They certainly simplify some things such as, issues of dissolution or circulation persistence due to differences in sizes. But I think that it also very much depends on the application. For example, having worked with people who conduct lithotripsy, for the most part, polydispersed microbubbles also have, very suitable applications. So.
All right, I actually had a question here. I was curious with regards to the neuroinflammation study. Do you recall if that was performed with, because you discussed bolus injections as well as continuous injections, and then there's this, there's some sort of dose dependence in terms of whether you have this neuroinflammation or not. Were the neuroinflammation studies performed with bolus injections or continuous injections or both infusions or both? And was there a difference in sort of this phenomenon?
That's a good question. So at least for our very basic neuroinflammation studies, these were bolus injections. And the reason was actually quite straightforward. It was just that we had no suitable solution to the fact that some of the larger microbubbles, like 6-micron microbubbles, would they would floatate out more quickly. So basically, as we were sitting there injecting these microbubbles, we had to just be aware of the fact that they would start to separate from the rest of the solution or the suspension. And so that's why a bolus was used in that specific experiment.
Okay, we have, I think, one more question before our next presenter from the audience here. Some microbubble manufacturers are unkeen on their imaging contrast agents being used as a therapeutic. Do you have any thoughts on how the community might encourage them to engage more enthusiastically with us, since our encouraging results don't appear to be enough? That's actually a really poignant and very good question.
That is, I mean, it's, this is a question that I've actually had to deal with myself when I used to work for advanced microbubbles. And, I think there are a couple of things. I'd actually like to leave the answer of that question up for discussion for the panel, which I think there are many people here that might have something to say about that. So, and I certainly will say something about that as well during the panel. So
I will certainly revisit and bring that question back. I agree. I think that's a good topic for discussion amongst the panelists. As long as you mentioned there are some regulatory challenges, when you add microbubbles to any treatment and therapy, and just sort of navigating through that minefield could also be a really good topic of discussion for the panelists. Brian, I think I'm going to pass the baton over to you here. And thanks so much, Dr. Song, for your presentation and your insights.
Thank you, Dr. Ford.
So thanks, Tyrone. Again, on behalf of the FUS Foundation and the AIUM. I want to thank all the speakers that were willing to participate today and the panelists. For our next presentation, I have a privilege of introducing Dr. Eleanor Stride. Dr. Stride is professor of bioengineering at Oxford University, specializing in stimuli-responsive drug delivery. She has over 180 publications, 7 patents, and is director of 2 spin-out companies set up to translate research into clinical practice. She's a fellow of the Royal Academy of Engineering and the ERA Foundation and the Acoustical Society of America. She received the Acoustical Society's Bruce Lindsay Award, as well as the IET AF Harvey Prize. Was nominated as one of the 100 most influential women in engineering in 2019. And was made an officer of the Order of the British Empire in 2021. Somehow, Eleanor, I feel like I need to bow at this point. So we really look forward to hearing your presentation and having a discussion with you at the end. She's going to be talking about pros and cons of cavitation agents other than microbubbles. We've heard a lot about microbubbles for therapeutic ultrasound. Eleanor.
Hello, and thank you very much to the organisers for inviting me to give this talk on the pros and cons of different cavitation agents. There have been many different types of particle proposed in the literature as potential cavitation agents, from nanobubbles to nanostars. And so I wanted to start by looking at what would be the characteristics of the ideal agent. First of all, we want cavitation agents to be stable, i.e. we'd like them to remain in the circulation for a sufficient time to enable a particular type of therapy. We'd like them to be continuously active during that time, and we'd also like them to be easy to handle and store prior to injection as well. We of course need them to be responsive to ultrasound. We don't want to have to use too much energy to activate them. We'd ideally like them to be trackable prior to activation as well, and we also want to be able to monitor them during therapy. Safety is essential. We need them to be made of biocompatible materials and to produce a consistent and predictable response to ultrasound so that we don't encounter too many regulatory hurdles in trying to translate them into the clinic. Similarly, we don't want to have to design completely new clinical pathways or hardware or develop new manufacturing techniques, as that requires huge amounts of time and investment. Within the body, we typically want to generate cavitation outside of the bloodstream, and so we need the agents to be able to extravasate and, of course, to remain responsive to ultrasound once they are in the tissue. And finally, we'd like to be able to functionalize these agents, for example, to attach drug molecules or targeting species to the particle's surface so that we can maximize localization of therapy. So how do microbubbles measure up against these criteria? Well, actually extremely well. They tick a lot of the boxes. They do have 3 major shortcomings, however. They don't last very long in the circulation, typically a few minutes in humans. Once they've been exposed to ultrasound, they are rapidly destroyed and they don't extravasate. So if we want to improve upon microbubbles, we need to look at the parameters we have to play with in order to engineer a better agent. Both the size and the composition of the agent will influence circulation time, acoustic response, etc. But if we focus on size to start with, if we want to improve stability and extravasation potential, this looks like the most obvious thing to change. And so, not surprisingly, there's been a lot of interest in so-called nanobubbles. I say so-called because, strictly speaking, they are sub-micrometer particles. They're not nano, but the term is now pretty used in the literature, so I'm going to use it as well. In terms of composition, nanobubbles are very similar to microbubbles, so they shouldn't present any problems in terms of biocompatibility. But their smaller size, and they're typically 10 times smaller than microbubbles, should mean they're less rapidly removed from the circulation. And indeed, there have been several reports in the literature suggesting that this is the case. These plots are comparing the circulation times for a micro and a nanobubble formulation, indicating that the nanobubbles persist for a lot longer. One thing to be wary of, though, is that in many of these studies, the concentration of nanobubbles is significantly higher, and as we've just heard in the previous talk, matching gas volume fraction is extremely important. It's also very difficult to be certain in vivo that bubbles remain in the sub-micrometer size range. And that's important because the efficiency of a bubble as either an imaging or a therapeutic agent changes dramatically with its size. This is a plot of the scattering cross-section of differently sized bubbles, so the ratio of scattered energy to the incident ultrasound intensity. And as you can see, there's a difference of many orders of magnitude between a 2 micrometer bubble and a 200 nanometer bubble. That means is that you need to use a much higher pressure to generate the same signal from a nanobubble. Or if you prefer to think of it in terms of cavitation nucleation, the minimum pressure amplitude required to activate a nucleus is the Blake pressure. And unfortunately, because the effect of surface tension, the blast pressure, increases with decreasing bubble size, again, you need much higher pressures to activate a nanobubble compared with a microbubble. This is also a good time for a quick side note on extravasation. It's been held until recently that provided particles are small enough to fit through the gaps in the endothelial lining, so between the cells lining a blood vessel, then those particles will be able to extravasate. There's actually been an increasing number of studies in the last 5 years suggesting that actually other mechanisms may be much, much more important in extravasation, and so we may need to think about how that feeds into cavitation agent design as well. But back to nanobubbles and how they measure against our criteria. Well, the good news is that we've addressed the circulation time challenge. They will also probably, well, certainly the literature suggests they are extravasatable, even if we're not completely sure why. The bad news is we've traded that for requiring much more energy to activate them. And we've also now got some potential issues with drug loading capacity and whether they pose any safety concerns as nanoparticles. So if changing the size isn't enough, what can we do with the composition of the agent? Is there some way that we can retain a good circulation time without losing detectability? And this brings us to nanodroplets. So these are again similar in composition to nano and microbubbles, but using a higher molecular weight core, typically a perfluorocarbon that's held in a superheated liquid state by surface tension before the particle is exposed to ultrasound and then vaporizes to form a microbubble. And the advantage of that is they are much easier to detect with conventional ultrasound imaging. As you can see here, the vertical white line is the activation pulse. And once the nanodroplets have been vaporized to form microbubbles, those microbubbles are very easy to image even at low concentrations. Now, sadly, it's not all good news. This time, we're trading off predictability. If we look at how microbubbles behave with increasing driving pressure, at low pressures, they oscillate in a very controlled, predictable way. But at higher pressures, we see much more chaotic behavior. And unfortunately, with nanodroplets, the pressures needed to vaporize them mean that we're always in that chaotic regime. Now, in addition to that, because vaporization is a stochastic process, it also gives us a problem with defining what exposure conditions should be used. Where do we define the necessary pressure threshold? If we want to ensure a sufficient number of bubbles are formed to produce a given bioeffect, then we actually need to use quite high pressures, but then the risk of off-target effects is significantly increased. Alternatively, if we define a low threshold for safety, then we lose reproducibility of the effect. And there's a further problem. The behavior of bubbles evolves over time due to the fact that they will interact with each other and form clouds, and those clouds may produce very different effects from single bubbles. If we look at the formation of a bubble from a droplet, over the first couple of ultrasound cycles, we get a single bubble that behaves in a nice predictable fashion. But by the time we get to the fourth cycle, we have a cloud that looks very different. So we not only need to worry about the pressures that we're using, but also the pulse duration. So if we sum up for nanodroplets, we've regained detectability, at least once they've been activated, but unfortunately at the expense of predictability. And we've also still got the problem that once the bubbles have been formed, they will disappear quite rapidly. And that brings us on to solid particles. It's been known for many decades that seeding a liquid with impurities will reduce the activation energy. And this is thought to be due to the fact that solid particles consisting of hydrophobic materials have nanoscopic crevices on their surface in which gas becomes entrapped. And that gas can then be released when a sufficiently large peak negative pressure is applied. And so several different types of solid nuclei have been engineered to deliberately incorporate hydrophobic crevices. I'm showing here the example of nanocups that I know Dr. Coviello is going to talk about in more detail later in this session, and then a high-speed camera video of the nanocups in action. Similar to nanodroplets, once a sufficient pressure is applied, then multiple microbubbles are formed that coalesce to form a bubble cloud. But the big advantage over microbubbles and nanodroplets is that solid nuclei enable cavitation to be sustained over much longer periods. As you can see here, microbubbles are destroyed very rapidly, whereas the nanocups enable cavitation over several minutes. So if we summarize for solid nuclei, they have lots of the same advantages of nanodroplets and the extra benefit of sustaining cavitation for longer, but they're still not detectable prior to activation, and they're not as predictable as microbubbles. There may also be some extra problems with functionalizing them, for example, to attach drugs or targeting species, because their surface properties are absolutely key to being able to generate cavitation. So where does that leave us? Well, I think the simple conclusion is there isn't one single ideal cavitation agent. Rather, we need to consider what features we need for different applications. The agent that is best suited for enabling delivery of drug molecules to individual cells is likely very different from that that's required for mechanical ablation for histotripsy, for thermal ablation, or indeed for generating chemical effects. And so what I think we need to do is think about engineering agents specifically for these different indications. So before I go, I must thank the many members of our team and also colleagues and collaborators elsewhere in the world, of course our funding bodies. Thank you for listening, and I very much look forward to the discussion.
Thanks, Eleanor. I really appreciate you presenting this. This is a very hot topic right now for a lot of different reasons. And so we're really interested to hear your thoughts about these types of agents and giving us alternatives to the microbubbles that are being pursued for therapeutic ultrasound. So we've had a couple of questions that have come through. Wanted to know if you could comment on the actual cavitation mechanism for solid particles that you discussed and why it is that they don't stop cavitating.
That's, very good question. Short answer is I don't think we absolutely know. If we accept the hydrophobic crevice hypothesis, that is, they are acting in the same way that native cavitation nuclei do, and that you have a region of enhanced surface energy where it's much, much easier to form a bubble. There are two possibilities. The first is that hydrophobic crevice remains intact and it can continue to entrap gas and so provide a continuous source of cavitation. The alternative to that is simply that we are able to inject much higher concentrations of solid particles than we are of microbubbles. We will only activate a certain proportion each time we fire. And so it may simply be that the probability of cavitation is much, much higher because we have so many more particles. And I think that remains an open question and further studies need to be done to tease that one out.
So if the application of nanodroplets is a drug delivery, such as drug release from droplets, aren't we simply interested in their vaporization? So is there, an issue with the sterility in terms of their long-term response?
I think that really does depend what we're trying to do with the nanodroplets. And again, it's what is good for drug delivery is not necessarily what's going to be good for thermal ablation or mechanical ablation. I think there is another challenge with nanodroplets, which is actually the quantity of drugs you can load. But again, you can inject more of them. So I think there are calculations to be done there. And I also think that it's not just the vaporization. Actually, I think where we are trying to have take-up of droplets, either through extravasation or active uptake by cells, actually that's really important before we hit them with ultrasound too. They're playing two roles in that case, and we shouldn't neglect that when we're designing them.
So you also mentioned the challenges of nanodroplets needing higher acoustic pressures. And you also mentioned large bubble breakup into clouds, which is a function, again, of the high MI driving. Can you comment on low boiling point PFC type nanodroplets as an alternative to avoid the high pressure complications?
You still need relatively high pressures. So, I think certainly both the Colorado and the Carolina groups, and also Toronto, have done some beautiful work mixing different fluorocarbons and showing that you can reduce that threshold. And we've been trying to do similar things too and optimizing the shell. You are still needing several hundred kilopascals to get vaporization and probably over megapascal to get reliable vaporization, compared with 100 or 200 kilopascals for microbubbles. So it is still higher. You are still definitely going to be getting a violent cavitation once that bubble is formed. I'm deliberately avoiding the use of inertial cavitation for reasons that I would love to discuss later. But yeah, you will be getting cloud, you will be getting uncontrolled cavitation once you vaporize them.
So how do the nanodroplets compare to nanobubbles with respect to circulation time, thresholds, and such?
It very much depends on the composition. I'm not absolutely sure we've got a good method for quantifying that yet. I think we know the half-lives are significantly longer than microbubbles, but I think we're still talking orders of magnitude rather than being able to precisely compare the two. I've seen estimates ranging from sort of a few hours to allegedly 2 days, but it does depend on the detection technique that you use. And I think, yeah, surface composition becomes much more important with nanoparticles than with microparticles.
What about combination agents? I mean, such as, putting bubbles together with droplets or particles together with nanodroplets and so on. Any thoughts on that?
Yeah, it's again a very good question. I know there were about 3 papers came out at a similar time trying to look at which came first. You need a bubble to cavitate in order to activate the droplets, or is it once you've activated the droplets that you then get inertial cavitation? I said it, sorry. It looks as though there is a sort of chain reaction effect. I don't think you need microbubbles in order to activate nanodroplets. We've certainly tried to purify them, at least as well as we can. We're pretty sure we've not got any microbubbles, and we see vaporization at similar thresholds. But equally, if you have some microbubbles present, it seems to accelerate. I don't think you need microbubbles to initiate it, but there may be some advantages in reliability. On the other hand, then you've got a much more difficult thing to explain to the regulators as to, and that's, I guess that's a topic of discussion.
Yeah, that was certainly one of the questions that came up, because in one of the statements here is that at a recent meeting, it was indicated that the US FDA is a bit twitchy with regard to nanoparticles. And some of the vernacular may be something that is kind of important to consider in these regulatory pathways. Do you have any other insights in terms of issues related to regulatory pathway?
Yeah, I mean, we're not doing it ourselves any favors. I think nanobubbles and nano other things are getting used as terms because they sound good for high impact journals. In most cases, we're not talking about nanoparticles at all. They are not less than 100 nanometers in diameter, and particularly nanobubbles, once they're injected into the bloodstream, they're expanding. And actually, I think we're really talking about the smaller end of a contrast agent. It's sort of the 800 nanometers, which probably means the safety concerns are quite significantly reduced. But we need to understand more about their pharmacokinetics. We need to understand how long they're staying in the body, what they're doing whilst they're in there. I'm afraid we've got to do more work. I think the regulators are right to be cautious because, as we all know, cavitation is a very violent process. And if we're introducing things where the predictability is dubious, then we're right to be cautious.
Okay. Well, in the interest of keeping us on time, there are a couple more questions. Let me do one more quickly. It said, if you had to choose a variety of cavitation agent for sustained bubble activity, what would you choose?
I mean, honestly, it would depend what I was trying to do with it. If I wanted to do a clinical trial tomorrow, I would go for an infusion of microbubbles. Just slowly making sure that we've actually matched the infusion rate of microbubbles to the flow rate through the tumor, and then we're pretty sure what we're getting and where it's going to happen. So that would be my immediate answer. I think there are very interesting things to figure out with optimizing it, but we've got to do some more lab work for that.
Yeah, so interval triggering, kind of like has been done with imaging agents in general,
exactly,
to maintain their activation. So, okay. Well, Eleanor, again, thank you so much. This is a lot of fuel for thought, and obviously we'll have our panel session coming up a little bit later today. So please stick around and join us. I'm sure there'll be more questions coming through at that point.
Fantastic. You didn't ask me who my favourite James Bond is, so.
Oh, okay, well, that comes for the panel discussion since we're at our magic time. So, Tyrone.
Yeah, I think we should hear from all the panelists on who the next James Bond should be. But of course, being that you've been knighted recently, you know, your opinion maybe carries a little more weight. All right, so our next presenter probably does not need much of an introduction. I think most of our presenters don't need much of an introduction, but with respect to opening the blood-brain barrier, Dr. Fleur Heinen is a pioneer. His research has focused on studying the effects of ultrasound beams on tissue and their utilization in therapy. He's investigating the use of focused ultrasound for non-invasive surgery, vascular surgery, targeted drug delivery, and gene therapy. I should actually also mention he's the vice president at Sunnybrook Research Institute, which is in Toronto, Ontario. He has developed theoretical models to help understand treatment parameters and to allow optimization of treatment devices. He has conducted in vivo experiments to investigate the various biological endpoints that can be induced by controlling the sonication parameters. His research on ultrasound transducers has been used to design and develop new phased array applicators and better ways to deliver therapy. He has been a leader in a variety of applications of focused ultrasound, in particular transcranial brain applications of ultrasound, devices that can be used for generating cavitation, mapping, monitoring, detecting cavitation, and then looking at those correlations between that cavitation activity and the resultant bioeffects inside the brain. So he's going to speak today on his presentation is on microbubbles as cavitation agents for blood-brain barrier opening. And we'll start the presentation and then have hopefully a very lively conversation with him.
Good morning, I'm Eleanor, and I'm going to talk about microbubbles as cavitation agents for a blood-brain barrier opening. Blood-brain barrier is formed by tight junctions that bound endothelial cells together in such a way that only very small molecules can enter into the brain. In addition, it has a biological barrier where efflux pumps up, removing any foreign molecules from the brain. So it prevents about 98% of foreign molecules entering into the brain. It's a major barrier for therapeutics. And the idea has been for many years with people to maybe use ultrasound to modulate that barrier. So this is now in clinical testing. This is one of our patients at Sunnybrook, where there is the tumor target. This is breast metastatic disease with HER2 positive tumor. And here is the cavitation dose that we modulate the blood-brain barrier with ultrasound and microbubbles. And this is the enhancement of radio-labeled Herceptin inside the tumor. You can see that we up to 450% more Herceptin has accumulated in the tumor. So it seemed to be working reasonably well in clinical trials to allow drugs to be delivered into the image-specified locations. So there has been now 20 years of experimental research exploring microbubbles and ultrasound in blood-brain barrier. And here you can see electron microscopy pictures of brain after sonication. So this is before sonication. You can see how the tight junctions are very well sealed. This is 1 hour after sonication. You can see the marker molecule is getting between the endothelial cells, the basement membrane. So the tight junctions have been opened. And here now 4 hours later, the tight junctions are starting to heal, and only part, the marker molecule is entering only part of the way of the tissue. So that is one mechanism. That is what we expected in the very first experiments. But we also discovered another mechanism. This is 5 minutes after sonication. You can see that endothelial cell has these small vacuoles that have the marker molecule inside them. And then 60 minutes later, those vacuoles are on the other side of the endothelial cell. So there seems to be an active transport that takes these vacuoles across the endothelial cells. And and here is a picture where the vacuole is emptying its content to the basement membranes. So we have also and others to have looked using 2-photon in vivo microscopy to see what's happening to vessels. And we have identified kind of 3 mechanisms what goes on. One is this fast leak that happens during the sonication and then stops after the sonication. Another one is where it's a relatively fast leak that then accumulates the drug, and then there is finally a slower leak that initially you don't see any molecules getting into endovessels, and then you see a accumulation of molecules in the brain. And when we look, compare the vessel diameter and see what the permeability is, you can see that the fast leaks mainly happen in the smaller vessels and the slow leaks in the larger vessels. And so it kind of agrees with the electron microscopy observations. We also looked into the impact of the sonications and microbubbles on the vessel microvasculature. And Dylan McMahon took brain samples and then outlined the endothelial vessels, microvessels, and took them and analyzed them. And what he found was that there is a transient upregulation of inflammatory-related genes in these vessels. So there is a transient inflammatory reaction. There was also the downregulation of blood-brain barrier transporter genes, and this means that the efflux pumps are inhibited, which is important for allowing drugs to stay in the bloodstream, in the brain. And there was also upregulation of angiogenic-related genes. And this is studied by others, Park et al., where they showed that oxytocin concentration in tumors with sonicated with ultrasound and microbubbles, the drug concentrations stayed the same after comparable 1 hour and 24 hours, whereas in tumors that were not sonicated, there was an efflux of the drug, and same way focused ultrasound in brain, normal brain tissue, there was much more drug right after the sonication than without sonication. And it also stayed there in 24 hours, indicating the efflux pumps are inhibited. And this is another showing kind of same effect. What about bubbles? Does it matter what bubbles we use? So most of the initial work was done with Optism, and then people were switching to Definity. And this is the first study comparing Optism and Definity, and they seem to work about the same. This is the probability of blood-brain barrier opening versus pressure amplitude. It looked like there is a little more enhancement with Optism than with Definity, but it's all in margins of error. So it seems like the standard imaging bubbles work. There are others have done more work on this, and Pauli was presenting last week about this, talking about different bubbles and different tests. So it looks like almost any bubble works fine for the blood-brain barrier opening. Well, does the size matter? We did lots of simulation trying to look what happens when we sonicate tissues. And here is the shear stress now on the wall. And here is bubble radius for different frequencies. You can see at 1.5, it looks like 3 micrometer size bubble. It has the biggest shear stress. And when you go to 1 megahertz, it's lower. And when you go to half megahertz, it's even lower, and looks like the bigger the bubble, the more shear stress we are introducing. And the same is true for transmural pressure. So Joy did the first study looking into bubble size. This was done at 1.5 megahertz, and from the previous picture, we need to relate that there could be a frequency dependence. So he looked two populations of bubble, small 1 to 2 micrometer bubbles, and then about 4 micrometer sized bubbles, and demonstrated there is an increase in fluorescent versus acoustic pressure. In the smaller bubbles, we have only a small increase, whereas the bigger bubbles, you have significantly larger increase in the fluorescent concentration in the brain. And they also looked histology, and they seem to be comparable histological observation. However, there were not extensive histological and transcriptome studies looking what the downstream effects might be. Then there was a study has shown that maybe it's not the bubble size what is important. This study was done 1 megahertz, 2 micrometer sized bubbles, and 6 micrometer sized bubbles. And again, 6 micrometers seem to be producing more blood-brain barrier permeability enhancement than the small bubbles. But when the results were normalized for a microbubble volume, they seem to correlate, be on the same line. So the bubble size is the same line. So from this paper, it looks like the explanation why the larger bubbles are more effective is really the gas volume. So if you have equal gas volume, then bubbles behave the same. So Dallin Mahan again looked into this effect. So he looked different bubble sizes and size distributions. These were he compared definitive bubbles. So here is now the bubble diameter, and here is the gas volume or account number. And he had definity and experimental bubble from Braco, and then 4 micrometer size bubble from Braco. And then he normalized all these experiments to gas volume. So here you can now see the second harmonic emissions after during sonications. You can see decay times and see that the experimental bubbles seem to last longer in circulation than the definity that is decaying pretty fast. And also the 4 micrometer bubbles initially seem to be less signal, but also it seemed to last longer. The half-life seemed to be similar to with the 2 Braco bubbles. So then he looked at contrast enhancement percentage and the exposure coming, the second harmonic emissions coming from the bubbles. And the bottom line here was that the percentage of contrast enhancement was the highest with the definitive bubbles. So when you compare the second harmonic signal, you got if the same second harmonic signal emissions, you get the highest enhanced with the definitive bubbles. And the experimental bubble and the 4 micrometer bubble were about the same. Then when he looked into this permeability and pro-inflammatory mediator concentrations, so here is the 2 log-fold change, and this is K-trans, so indicating what is the amount of permeability change, and looking at different pro-inflammatory mediators. They all showed similar trends. They showed that definity and the polydispersed experimental bubble had about similar K-trans values and similar pro-inflammatory mediator activation. Whereas the 4 micrometer size bubble induced bigger pro-inflammatory mediators chains with a smaller change in the K-trans values, meaning that we were able to cause smaller probability enhancement while increasing the impact on these inflammatory factors, which might be important when we are looking to induce therapeutic effects in the brain that are not necessarily related to blood-brain barrier. So interesting observation. Others have looked even smaller bubbles. These are lipid nanobubbles, about 200 micrometers in size. And the interesting feature of these bubbles that in vivo, they seem to not have the typical signal pass curve that other bubbles have, like Definity or Optism, these curves, but they seem to be a leveled approach. So I don't quite know why that is, why it doesn't show the signal pass. Maybe the bubbles are staying pooling in lungs or heart or somewhere and then slowly be released. And that's why you get this nice flat distribution. These are acoustic emissions coming from the bubbles. They seem to be as effective or maybe more effective, and it's Definity or Optism in producing blood-brain barrier opening. You can see these enhancements here with all of them. So even these very, very small nanobubbles seem to be effective in modulating blood-brain barrier permeability. They produced some histological evidence, but the study was not completely thorough. So it's not quite sure if this is absolutely safe or not, but seem to produce a good blood-brain barrier opening. The final thing that I like to talk about is acoustic cluster therapy, where microbubble is bound to droplet, and this is electrostatic attraction, so they float together. This is a superheated droplet, and they are injected in the bloodstream, and they float together. And then when they're exposed to ultrasound, the droplet is vaporized, and then this big bubble that's bigger than the diameter of the vessel is then sonicated to enhance drug delivery into the brain. And here is an example, so demonstrating blood-brain barrier opening immediately after sonications. So these seem to be working quite nicely. It's still open at 1 day, although at less, and 3 days seem to be completely healed. They also did histological studies which didn't show any significant amount of tissue damage. However, they didn't produce long-term studies or repeated treatments and so forth. So again, interesting study showing effectiveness, but I would like to see more data showing safety. So as a summary, almost any bubble with ultrasound can enhance the blood-brain barrier permeability. And this is now in clinical testing, which the standard ultrasound polydisperse bubbles, ultrasound diagnostic polydisperse bubbles. And most experience is with Definity, and lots of animal work has been done on lots of clinical work, maybe not lots, but some clinical work, but other bubbles seem to work also. And there is evidence that nanobubbles and even larger bubbles are working well. There seem to be induction of pro-inflammatory mediators that correlate with K-trans, and the bubble size seem to have an impact on the enhancement. Larger bubbles seem to be better in the enhancing more, and this might depend on gas volume. And large monodisperse bubbles might be more effective in inducing pro-inflammatory, enhancing pro-inflammatory mediators. So that's it, and just want to acknowledge my group and funding and collaborators, many of them. Thank you for listening.
Oh my goodness, how do you manage such a large group? That's like a company. That's the first question.
Not very well.
So we do have a few questions that are in the Q&A. So the first one I have here, first and foremost, thanks so much for the talk. Really valuable insights there. Do we know if the vessel size dependence on slow and fast leakage is dependent on the bubble size?
Yeah, so I don't know. We don't have experimental evidence. There is some simulation data that might indicate there is dependence on the bubble size and relationship between bubble size and vessel size. But how strong that is, I don't know.
So a follow-up along the same lines, and maybe there isn't experimental evidence yet. Is there a difference in the time for resealing between small and large vessels post-BVB opening?
So I don't know the answer to that either. The fast leak seemed to seal like really quickly. So the slower leak seemed to last longer. So my guess would be that the small vessels seal faster than the large vessels because that's where you get the slow leaks. But we haven't extensively studied this. Those two photon microscopy experiments are difficult on long time frame because of need to keep the animal stable, and it's not a trivial task.
Yeah, I can see that, or I can appreciate that. The next question I have here is, commercial echo contrast agents are approved for very specific clinical indications, and the route of injection is limited to IV. Do you think these agents could be delivered on the arterial side, so intraarterial injections, without concerns in the brain?
So that's a very good question. I definitely think that there is a concern if you inject intraarterial. You would think that you would like to inject them intraarterial, especially in some cases where, because you can get 10 times more agent into the brain, same bubbles. But I think the large bubbles potentially could cause microstrokes in brain. So at least the polydispersed bubbles, they are filtered by the lungs, and that's why they are so safe in brain. So I wouldn't inject bubbles intraarterially. I would inject the therapy agent intraarterially in some cases where you cannot have enough volumes, like gene therapy, maybe.
A follow-up to that, what do you think about the nanobubbles? Could you potentially inject the nanobubbles intraarterially?
Yeah, that could be potential. There is very little data. There's the 200 nanometer size. Yeah, there is not too much data. Some of the data I have seen looks very promising, but it's not complete. So even intravenous injections, I would be hesitant to use them in patients now. I would like to see much more animal work done. And like I said, that the curves do not, the bubble response curves don't seem to be the same what you would expect. So there is something funny with the circulations and what happens. So lots. It's very exciting, but we need to do more work with the polydispersed commercial bubbles. We have 20 years of experience with the hundreds of labs, so there's much more experience.
That's a really good point with regards to the nanobubbles. And there's a follow-up actually with regard to nanobubbles. With respect to their circulation, with the differences in particle recognition to systems like the RE, a system be different and possibly explain maybe the differences in like the bubble response and whatnot?
It could be. I don't know. I don't know the answer to that question, but it's a very good question. So like I said, we need to do many more studies to know.
So in your opinion, you've worked for a number of years now with opening the blood-brain barrier, and you've explored a range of parameters. So in your opinion, which one is most important? Transmitted frequency, pressure, bubble dose, bubble size, in order to get a safe opening of the blood-brain barrier?
Again, a good question. So, right now the commercial bubbles are the ones that we are using in clinical trials. They seem to be safe. In order to go to the skull, you want to use low frequency. The low frequency seem to be more, multiple reasons. So number one is that the focal spot size is large enough to see some effects. Because the higher frequency, then the focal spot size is so small that you spend for the whole day to try to paint over the tumor. The second thing is of course skull propagation, and you want to be low frequency to not to get the sound. You can get enough sound at a higher frequency, but then that you will be able to listen the signals coming from the bubbles. And that is key for the safety. It's absolutely key. But pulse parameters, you can have longer or shorter bursts. It doesn't matter. Bubbles, in my view, is the unexplored field. There is still lots and lots of work there, and I think that the results that are coming now from labs seem to indicate that the bubble really matters, that you can do lots of different interactions in the brain, depending what bubble you use, what kind of bubble. And so you could imagine, like, I think there's some evidence now that like Alzheimer's disease, we can reduce the amyloid load, cause this immune response against the amyloid. And that might not require blood-brain barrier opening. Maybe better to use a bubble that doesn't cause permeability change, but causes this inflammation response, like I saw it with that 4 micrometer bubble. So I think we need to understand much better the physical forces and how they translate to the downstream biological effects. I don't think we have a good understanding of that yet, and I don't think we have a good understanding on all the possibilities what we can do with ultrasound microbubbles in the brain. So there are many different therapeutic options there, not just drug delivery in my mind.
Okay, thank you so much, Kullervo, for your presentation and your insights, and looking forward to your participation on the panel. In the interest of time, we're going to shift to, I believe this the last presentation for the session. So Brian?
Yeah, so it's my pleasure to introduce Dr. Christian Coviello. He is co-founder and chief technical officer for Oxonics Therapeutics. He leads the development of Oxonics' proprietary system, Sonotran, designed to enhance anti-cancer drug delivery into solid tumors, which is entering its first in human clinical trials starting this year. In this role, he leads a team that covers both preclinical development and analysis, along with hardware design, including novel multi-system probe and different components of that, as well as proprietary design of software. So He has a special interest in applications in monitoring of cavitation-enhanced focused ultrasound therapies, a subject which he's authored and co-authored several papers and has had grants along with patents. So, Dr. Coviello, we look forward to hearing your presentation and spending a little time in the Q&A with you.
I'd like to thank the Focused Ultrasound Foundation, as well as the AIUM and the organizing committee of this workshop for inviting me to speak. The problem that I'll be speaking about is improving drug delivery in oncology. And the clinical need that we're trying to address is that of many drugs have very poor delivery into solid tumors. So one example, this is in the case of head and neck cancer, and this is looking at the accumulation of an anti-EGFR antibody. In this case, the commercial name is Vectibix. And where you have patient slices of tumor, you see stained here in brown the cancer cells. And where, if you look under fluorescence imaging, where you see the drug, the Vectibix, it's mainly around the periphery. If we then look at staining for the vasculature, we mainly see the vasculature at the periphery. There's very heterogeneous intertumoral antibody accumulation. The accumulation is mainly at the periphery of the tumor mass. So poor distribution, but also poor penetration. And the way that Oxonics is trying to combat this problem in delivery of drugs into solid tumors is, this is a cavitation agent workshop. Well, we're trying to use our own cavitation agent, the sonotram particles, that when excited with ultrasound, create a microstreaming micropumping effect. And you can see an example here in a static well chamber, where we can cause a stream on the order of tens of microns away from an individual particle. For particular reasons that I'll go through later in this talk, we developed our own ultrasound system so that we can ultrasound image to find solid tumors. We can then deliver focused therapeutic ultrasound to cause cavitation from our cavitation nuclei, to cause this micropumping, microstreaming, pump more drug. And then if we take whatever it is the standard of care drug for that patient in a completely unmodified sense. So into one arm, you'd get an infusion of our particles. Into another arm, you'd get whatever the standard of care drug. It's unmodified, completely unbound from our particles. Regardless of the class of drug, whether it's virus, antibody, ADC, or small molecule, we should be able to pump more of it into solid tumor. Now, our particles shown here on the TEM image are cup-shaped particles. You may have heard of them referred to in the academic lab from the University of Oxford, which is spun out, referred to as nanocups. Well, each cup-shaped particle is approximately 450 nanometers in diameter, with a 200 nanometer cavity, each cavity supporting or stabilizing a single air bubble. They're quite monodispersed. They have a very low polydispersity index. And the product is delivered to the clinical sites, so a pharmacy and a hospital, in a sterilized, sealed vial. So, and that's then compounded into a standard infusion bag, which I'll come back to later. So the cavitation behavior from our particles, this is work by James Kwan and colleagues using the Brandera system. You can see it has quite interesting cavitation behavior. You see some collapsing, you see some sustained collapsing, and you even see bubble-to-bubble interaction. This is in a very dilute sample. Now, be it far from me to say whether this is the behavior is purely inertial cavitation, whether there's some stable behavior. I'll leave that to Eleanor Stride's talk, which you saw earlier, for her to conclude. She's more of the bubble expert. But at least in the case when we look at this under flow conditions, which are somewhat biologically relevant, so this is in a very narrow flow channel where we're increasing the sonication pressure to cause cavitation from the particles, and we're listening from a separate PCD at 7.5 megahertz. What we initially see at very low levels of sonication pressure, or what we see in the frequency spectrum, we see some small harmonics. But then when it actually rises above the noise level, what you see is domination by broadband emissions. Now, is that entirely inertial cavitation? Well, I'm sure, as Eleanor said earlier, it's quite a bit more complicated. But generally speaking, we would say broadband dominated emissions. Now, interestingly, when you then compare this is some work from James Kwan here on the right, very early in the development of these. He made several formulations with different cavity sizes and then experimentally collected the cavitation threshold for those particular formulations and compared that to, as Christy Holland spoke last week, to a theoretical model of cavitation threshold behavior. And what James was able to show is there's very good agreement between what we call now our sonotram particles and theory in terms of cavitation threshold behavior. Also, in terms of some other ultrasound functional characteristics, the particles themselves, probably not surprisingly, don't really have much acoustic contrast. So in the left here, the two different flow channels, the top one was sonoview microbubbles, the bottom was sonotram particles, using a conventional Philips ultrasound imaging system, where we've tried to normalize the number density of particles or microbubbles. You don't see any acoustic contrast, and that's not surprising considering their size. But one of the main reasons then on the right is why we developed the particles back in the university days is our early animal experiments trying to create sustained levels of cavitation during a mouse experiment and trying to deliver a drug was very difficult. In this example, you see we had to do 8 injections. That's the arrows there, 8 injections to get a sustained level of cavitation. But then similar time length of treatment, but a single infusion of sonotran particles, we're able to sustain cavitation levels throughout the experiment. So that was one of the main reasons why we came up with them, was sustained cavitation behavior. The second reason is that we can actually pump the particles as well as drug out of the vasculature. So in this particular example, where you see cancer cells in blue, blood vessels in green, and our cavitation nuclei, which have been fluorescently tagged in red, we actually see only a few particles out of the vasculature into the tumor tissue, most of them actually occurring within the vasculature or the near perivascular space. But when we apply ultrasound here on the right, we actually are able to distribute them throughout the tumor. And that's what we would hope to do as well with the drug. So we can more evenly distribute both particles and drug throughout the solid tumor. And as we had to take that at the initial stage when we spun out the company, the formulation from a very small scale up to something that can now be used in the clinic, well, first we had to find a manufacturing site to be able to do that with the proper accreditations in place. We have a contract manufacturer that we use that's both EMA and FDA, as well as MHRA accredited. We then had to work with them to develop a GMP compliant process to manufacture them, as well as finding suppliers that can supply GMP material, or where we've audited those suppliers to ISO 13485. We also had to scale up the process. Initially, we were only able to manufacture 40 milliliters at a time. It was very slow. We now can manufacture at up to 20 liters, and that could be scaled further or even parallelized to scale up and out. I talked about the form factor of those vials. One of the things that you want to do in designing a product is you don't want to move outside of kind of normal clinical practice. So those vials are easily amenable to compounding within a hospital pharmacy into a standard infusion bag and giving set for then infusion into the patients. That's quite important. As well, we're a commercial outfit. We have intellectual property around the particles so that we can fully commercialize this. And then in terms of the regulator, we have a full GLP toxicology package that the MHRA have now completely reviewed. There's zero bioburden, there's no adverse findings in that package. Now, I talked about the particles. I have to talk a little bit about the system so that the next parts of this presentation make a little bit more sense. But we developed our own ultrasound system. And one of the main reasons why we wanted to do that was to be able to get the best effect out of our particles. Quite uniquely, this system has a multifunction probe. And by multifunction, I mean it does B mode imaging to guide the treatment. And out of the same aperture, it sends in, so in a curvilinear handheld form factor, it sends in therapeutic ultrasound, focused therapeutic ultrasound to cause cavitation from our particles at 500 kilohertz, as well as it has a separate set of elements that can listen in real time for the acoustic emissions from the particles and map that. Now, you may call that passive acoustic mapping, which is, you know, what we had called that in Oxford, or you can call it passive cavitation imaging, which others use in the community. Makes no difference to me. But it's an important part of our product that we can be able to monitor that treatment to know where we're causing cavitation in real time. In addition, one of the other unique features of our system is that we can live register ultrasound to pre-collected CT or MR. And that's quite a unique feature, not only in terms of improved guidance of the treatment, but also in terms of knowing where cavitation events occurred relative to 3D anatomy. So we can see volumetrically where we cause the effect and does that correlate to efficacy, which I'll talk about a little bit later. As well, we developed our software with clinicians so that it was easy to understand and easy to use. Now I talk about coupling cavitation to efficacy, and Cameron Smith gave a great talk about this last week. But one of the things that we would like the community to continue to push is coupling cavitation dose to some kind of effect. Here we see an example, where we're causing PAM. This is in a murine tumor as we're treating. And in this particular case, we were trying to deliver anti-PD-L1 antibody to these mice. And I highlight in the stars here two different cases, one of a good responder in blue and one of a poor responder in red. And what we see in the poor responder anecdotally is that the cavitation events in terms of the little dots there, dots in terms of their spatial location and size in terms of the amplitude of cavitation, we see mainly very low cavitation emissions, as well as mainly being on the periphery of the tumor. Not surprising, a poor response in that case. And in the case of the good responder, where we saw complete regression of that tumor, we saw large amounts of cavitation, but spread distributed into the center of the tumor. So not surprisingly that we got a good response there. So quite important in terms of, in Cameron Smith's talk, coupling that to numbers, that's something we would like, Oxonics and others to continue to push forward within the community in terms of cavitation dose coupled to effects. Now getting back to kind of more system level testing of our Sonotran particles with our Sonotran system and showing first the infusion rate coupled to sustained cavitation levels. We tested this in 8 pigs where we were treating regions of the liver and the pancreas. And what we were able to show is we could sustain cavitation for upwards of an hour from what our estimated standard human dose is. So here you see on the top right, that's my hand sonicating this pig's liver. We're focused, see on the bottom here at that green crosshair. In the bottom is before we infuse our particles, but are sending in therapeutic ultrasound. And then once we actually start to infuse our particles, we almost immediately see cavitation represented by the color map using our passive acoustic mapping algorithm. Most importantly, coupled to the sustained cavitation behavior, we didn't see any adverse events associated with this treatment. And that's highlighted in post-treatment histology, where there's no real difference between control tissue and actually treated tissue using cavitation initiated from our particles. I can't really dwell much on this, but we have a comparable data set across a range of macromolecular therapeutics, including viruses, antibodies, and ADCs. And the general conclusion is that if you get more of the drug in, you get a more efficacious response, either viral activity leading to tumor regression, increased dose leading to improved median survival. So in conclusion, we developed the Sonotran particles specifically for their ability to enhance drug delivery. They have a unique size, morphology, as well as unique cavitation characteristics. We've done an extensive amount of development from the academic days to be able to scale up, to be able to manufacture them to GMP, doing all the pre-clin talks, all the safety validation testing, as well as a long amount of regulatory documentation to support their clinical use. And combined with the Sonotran system, we have an extensive pre-clin data package which shows their effectiveness in improving delivery of a range of oncology agents. And we will soon to announce our clinical trial in this year. So I have to acknowledge all my colleagues at Oxonics. There is a laundry list there to the left, as well as our colleagues at the University of Oxford, foremost our co-founders and still consultants with us, Konstantin Kusios and Bob Carlisle. Thank you very much.
Thank you, Christian. This is a great talk, and we really appreciate the opportunity to hear kind of what I would call a very advanced development project for this, and yet trying to push it toward the clinic and being very different, not only in terms of the materials that are being used, but also the ultrasound systems that are being used. So a few questions that have come through in the system here. So the first one is, it looks like in this, kind of in keeping with the theme of this session, it looks like the results in slide 11 really point to the need for the cavitation dose. This is where you saw a good response in one case and a not so good response in the other. So what exactly is being measured then in that circumstance using the passive cavitation approach that you're using?
What we're measuring in situ on a per pulse basis is the acoustic emissions in joules. And then we look at that over time as well. What we're hoping to do in our clinical trial and into the future is based on the distribution of those cavitation events spatially, as well as the total accumulated energy of cavitation, is to predict efficacy for those patients, depending on the amount of volume to be treated. So the number of lesions that were targeted.
Okay, another question that came through in the context of getting consistent over time cavitation, can you comment on using injections like bolus versus infusion of microbubbles and how that relates then to the infusion or administration of Sonotran?
Yeah, so that's a very good question, because when you go from kind of preclinical models with mice, I mean, definitely a bolus injection of microbubbles is pretty close to, especially you hit a saturation point, and so the bolus injection is kind of close to an infusion. But when you go to humans, obviously, it's a slightly different story. So we use a relatively slow infusion of our particles over time. That's most likely the reason, well, one of the reasons. I mean, you saw what Eleanor presented earlier as to there's quite a different, PK for nano size cavitation agents versus micron size cavitation agents, but the stability of our particles in bag, to be able to then be able to treat for the length of time that we need to treat, seems to be, at least from what we've done initially in the early days at the university. You saw the bolus injections of microbubbles in mice, how many we had to use to get kind of similar levels of cavitation over a duration of a similar time. And that's what pushed us to develop the particles. So it's, our method of treatment is that, from the start of infusion, we've got about an hour of total treatment time where we can cause sustained cavitation at a sustained level. And that's primarily due to the PK, but as well, just the number density of particles in the blood.
Another question that came through, which I think is rather interesting in the context of the cavitation dosimetry, which is sounds like from what you're describing and the images that you showed that microstreaming is a critical mechanism involved in this process, and yet we don't know how well microstreaming is related to the bubble activity. So can you give us a feel for what the relationship would be there in that case?
I'd love to, if only I could. No, I mean, definitely. I mean, we know that, through enough control studies that it's clearly the cavitation events and what seem to be at least dominated by inertial cavitation, as far as the acoustic emissions, or at least our understanding of the acoustic emissions at this present time, that are causing active transport of the drug. Now, because of the size, or the initial size of the bubbles, it would suggest that it's taking multiple bubbles and perhaps then bubble coalescence to be able to push things somewhere. I would suggest that is probably microstreaming. It would be very difficult, I think, to set up an experiment that was believable to where you could actually image that, but not have it be affected by like a single bubble kind of stream. So you saw the example that I showed in that's very artificial in a static well plate, where you get a stream that's on the order of 100 plus microns away from a single particle. Is that what's actually going on when you get multiple particles cavitating? It's definitely a lot messier than that, but it's not as though the particles themselves are imparting momentum transfer. It's got to be the bubble activity that's imparting momentum transfer to the therapeutic.
Okay. And so one last question before I pitch it back to Tyrone for the break. How different do you think, you mentioned that the high-speed videos that you were showing that there was a difference in the bubble behavior. How different do you think that bubble behavior actually is for these type particles versus, say, ones that are nucleated from submicron bubbles or droplets?
Yeah, so no, it's interesting. I mean, one of the main things that's quite different about our particles is that solid core that is probably one of the main reasons why we mainly only detect the acoustic emissions probably once the bubble has detached from the particle. So it's probably likely that there's some stable oscillations on the core, and then it actually detaches, and then you actually can detect cavitation from it. So the axisymmetric, the non-symmetric nature of the particle is probably affecting things. I think the nice thing, at least from what we've seen from manufacturing these for a while now, is that we get quite reliable cavitation activity and quite reliable activation of them. So the spectra that you see and the cavitation threshold behavior is very well characterized from our experience. My experience with droplet behavior is that it can be, at least in the past, it requires somewhat more energy put in to be able to get the vaporization. But the threshold behavior and the spectra behavior can be quite variable. Now, I don't mean to disparage these agents. I think they have their place, but at least what I believe, again, from working on this for quite a while now, is that the cavitation behavior from solid core particles like ours is quite regular.
Okay. Well, thank you very much again for a wonderful presentation. Like I said, this is kind of cutting edge stuff, and yet trying to translate it as quickly as we can to the clinic, which is what we're all very, very interested in doing. So thank you again. So I'll pitch it back to Tyrone.
Okay, so that was our last presentation. We're going to take a 10-minute break, get organized, bio break, refresh ourselves, get more coffee. Thank you, Eleanor. Tea. You are in England, so maybe drinking tea, English breakfast. And then we're going to return and we're going to have a conversation with our presenters as well as some panelists that we'll introduce once we return from the break. And so let's just take that 10 minutes and be back. That will put us at 11:50, 11:51 Eastern time. There's a whole variety of times for everybody that's participating in this, but we'll see you in about 10 minutes.
Yeah, so I'm introducing the session. So I just want to describe that we're going to be doing the panel discussion here. We'll have some opening remarks from those that haven't participated thus far or presented. We want to get to some of the targeted discussion questions that some of which have been submitted, and you can continue to submit questions and we'll monitor those questions as we go. And then a few concluding remarks at the end. So I'd like for Tyrone to introduce the additional panelists that are joining us in addition to those that have already made presentations this morning.
Okay, so we welcome our panelists who are experts in their own right with regards to contrast agents, cavitation agents, diagnostic and therapeutic ultrasound. We have here with us Paul Dayton, who's a professor and chair at the University of North Carolina, Chapel Hill. He's also a faculty member of the joint program, Department of Biomedical Engineering, jointly between the University of North Carolina, Chapel Hill and North Carolina State University. We have Christy Holland, who's a professor in the Division of Cardiovascular Health and Disease, the Department of Internal Medicine, the College of Medicine at the University of Cincinnati. We have Alexander Sasha Klubanov, who's an associate professor in the Department of Biomedical Engineering at the University of Virginia. We have Alfred Yu, who's a professor of electrical and computer engineering and a member of the biomedical bioengineering program at the University of Waterloo. And then lastly, we have Michael Caney, who's a scientific director for Carthera, which is developing a system for transcranial ultrasound therapeutic applications. And so I want to start off with Paul. We're going to go around the horn just to get some initial thoughts with regards to what has been presented so far from our presenters. So please, Paul.
Yeah, thanks, Dr. Porter. Yeah, it's been a great session so far this morning. We talked about pros and cons of different agents, heard a little bit about applications. And sort of what I heard is that there are really important parameters with any sort of cavitation agent. We talked about size, the benefits of microbubble agents that contain more gas volume. We talked about the benefits of monodispersity or reduced size distribution. We talked about small agents, which could have the potential to extravasate. We also talked about pharmacokinetics, how long different agents would circulate. And there are certain advantages to having an agent that clears quickly. There's certain advantages to having an agent that provides sustained cavitation over a long time. We talked about cavitation or activation threshold. So how much energy a microbubble or cavitation agent is going to produce when it's hit by ultrasound. Do we want this to be all the time? Do you want it to be always acoustically active, or do you want an agent that perhaps is not acoustically active until you reach a certain threshold? Now, when we talked about the potential of drug loading, do we want, how much drug do we want to be able to get in this agent, or perhaps not at all, perhaps just it's co-administered. And then, of course, there's characteristics of the shell. They might dictate pharmacokinetics, allow agents to localize in a certain area or affect the clearance. And the message that I got is there are a lot of opportunities for different things. A nano-sized agent might be really beneficial in certain applications, but a larger agent might be more beneficial in, let's say, blood-brain barrier opening or something. And the big disconnect I see is right now we only have a few options commercially right now to explore, and they're mostly, designed for imaging, right? You look at the microbubbles that are out there, and they're all microbubbles, right? There are no non-microbubble agents that are FDA or European approved or Asian approved right now. So we're limited with a few agents that really aren't designed for therapeutics. So I think there's a real big opportunity out there for all of us to continue to push towards new agents that are optimized for therapeutics with different characteristics. I don't think there's one size fits all. And there's certainly going to be different opportunities depending on the application. So that was sort of my take-home message of today. Happy to extend that for the comment and thoughts.
Thank you so much, Paul. Next up, we have Christy Holland from the University of Cincinnati.
Thanks, Dr. Border. So my take on this is my personal interest is in ultrasound-mediated drug delivery, even on the arterial side. There's tumors that are fed by arteries. There are clots that are in arteries. And so I was the one that asked Dr. Hinnanen that question about arterial route versus venous route of infusion. And so I think this is a kind of an important question, is route of administration, and can we get away with putting a nucleation agent into the arterial side, particularly if you want to sustain that bubble activity or you have sustained nucleation. I think we do want to bypass the lungs. I have particular interest in lytics, in anti-inflammatories, as well as bioactive gases. And it would help if we could move towards arterial administration, but of course we want to do that safely. And I appreciate the comment by Dr. Hinnanen about safety in the brain.
Okay, thank you. So next up, we have Sasha Klibanoff from University of Virginia.
I really enjoyed being on this meeting, and really very important points have been raised regarding the size of the particles and concentrations. And I think actually it is much easier than was just mentioned here to go and do it. All we need to do is take a vial of DEFINITY, turn it upside down, and set it on a solid surface for 15 minutes, then take half a ml out from the very bottom, and you have very small bubbles. So you don't worry about lungs anymore. So there's nothing to quote unquote filter about. And we just need to start doing these things. All it takes is just go and do them. Another thing that matters is gas exchange. This has been mentioned before, and it matters a lot. So gas exchange happens in the body, of course, and bubbles can grow and shrink. Sometimes, if they're too big, they might actually clog the capillaries if they're too big, and we don't want those large particles in there to clog the capillaries. By the same token, we can avoid doing that if we first dilute the bubbles into the media that has plenty of dissolved air in saline before the infusion, and automatically you have essentially no growth anymore, because there's air in those bubbles already at some amount. Yeah. We just need to start doing it.
Yeah, just do it. I've heard that tagline somewhere before. All right, so next we have Alfred Yu from University of Waterloo. Please, your perspectives.
Yeah, thanks, Tarang. I'm very excited to hear about various advances today on bubbles, or to be more generic, nucleation agents. On various fronts, we have the basic science. There's also a lot of activities going on in the clinical translation of new agents. From my perspective as an engineering scientist, I actually think we still have lots to be done, right? It's not only just about the chemistry and the physics. The cell biology and the interfacial biophysics are also very important. And as one of the speakers mentioned earlier as well, the notion of nanobubbles is still a bit of a hype right now, in the sense that we aren't really truly developing nanobubbles yet. So that's kind of like something which we should look further into. And so overall, it's very like stimulating. I have tons of ideas that I will try out later on. Thank you.
Yeah, maybe the host organization could provide some seed funding for all of us to work collectively to respond to Paul Dayton's proposal earlier, which was, designing these agents specifically for therapy.
And Tyrone, you can be the PI for this. You can lead this consortium, right?
All for it. So our last panelist to comment here is Michael Caney, Scientific Director for Thera. Please.
Sure. Hi, everyone. I think we heard a lot of fantastic talks today. I think that, from a therapeutic kind of commercial perspective for blood-brain barrier disruption, at least, I think that the microbubble is going to be one of the next maybe big areas where we can potentially innovate. From our side, from a company side, I can tell you a little bit. We're actually using implantable ultrasound with our SonoCloud device. We've done blood-brain barrier disruption in human patients with both SonoView and Affinity, and we've seen pretty similar effects as far as the magnitude of blood-brain barrier disruption. We switched primarily for commercial reasons, not any safety or efficacy reasons. But I think that the agent is really something that, could definitely be optimized further for therapy. I think that the big question is really like, what are the real advantages of switching the agent? I mean, the regulatory burden, the costs of commercialization, all of these things are huge. And I've been going to acoustic conferences for 20 years now and heard about different contrast agents and optimization. And it seems like none of them has made them across that bar of, having commercial potential. And I think the same is true in blood-brain barrier disruption. We have to say, what are the real advantages of switching the agent? Are we really getting a better safety profile? Are we getting more drug there? I think that has to be a pretty significant gain to get one of these agents across that hurdle. So I think that's one of my things. But I think it's an exciting field, and I think it's maybe the next thing that's going to be really looked at if this field takes off.
And perhaps we can have more sessions or contributions, participation at some of these conferences where that actually is a topic for a session, right? Just trying to navigating the regulatory hurdles, the pros and the cons for different agents, because we definitely have very thorough conversation around the science, but ultimately, if you want to get this to a clinical application, there's other things that have to be considered. Right. And so having that as part of that larger conversation, I think would be extremely valuable. So we're starting that, I think, here today, but I think it needs to happen more frequently. So I'm going to, Brian, if it's OK, I'll post the first question here because I got one that's just burning in my head. And I to some extent, blame Paul on this. And so maybe he'll be one of the first ones to offer up a response, but definitely this is posted to all of the panelists. And then we also will return to some of the questions that remain in the question and answer queue. But the first one, I'm with you 100%, Paul, with regards to the agents, the new cavitation agents that predominantly have been discussed that are approved clinically or designed for diagnostic applications. They were designed, I want to repeat that, they were designed for diagnostic applications. So one of the questions we did post to you all early for consideration was what should be considered when designing a contrast agent or a cavitation agent for therapeutic applications? What are some things that should be considered? And this is for the entire panel, please.
Yeah, anybody can jump in on this one.
Including the speakers.
Well, you want a sustained cavitation, Tyrone. So it's not just about the threshold, the whole discussion we had last time. It's about exceeding the threshold. And something that has surprised me, and Christian's data kind of spoke to this too, is that we all kind of, inertial cavitation used to be called transient cavitation because we thought it was temporally transient. However, it turns out it's not true. You can sustain inertial cavitation over a longer period of time. And so I do think that that's what's special about what we're all trying to do is, can you target a particular part of the body that then causes this bubble activity over a sustained period of time? And a lot of these discussions thus far have been focused on the bubbles. I think it's time that we start focusing on the bioeffects. What exactly is going on? There needs to be some basic science to understand how the cells that line the arteries and the veins interact with these bubbles. What is the biological response? What kind of proteins are involved? What kind of genes are turned on? And probably very few of us that are on these squares that I see on my screen are going to answer that question. And I think we do need to partner with people that are trained to figure those kinds of questions out.
Yeah, just a quick comment there. I mean, the field of mechanobiology, right, has really sort of ramped up and taken off, right? And so there might be some opportunity to partner with people who are experts in those areas, along with like transcriptomics. We saw some data on transcriptomics earlier, but Eleanor, please.
No, just on mechanobiology, I think it's very interesting though, because they tend to be working at much lower rates of stimulation. I mean, so we talk quite frequently, but it's funny, they were like, oh yeah, we're doing this at high frame rates, 30 hertz. And so I think that's quite a significant challenge for us, as we're talking microseconds, sub-microseconds in many cases for the stimulation of very high frequencies. And no one knows. So I think that's great for research. It's not.
Yes, you can really begin to start challenging. You can really begin to start challenging people that are thinking in terms of different scales, different time periods, but really challenging them to think differently about these kinds of questions.
I'd like to attract attention to the geometry. Dr. Holland mentioned arteries, etc. But I somehow think that what matters is what's happening in the capillaries, where the bubble is actually against the wall, right next to endothelium. And in all the other places, bubbles are, many of them are in the bulk away from, all they touch is red blood cells. So just want to make sure obviously capillaries are short and passage through the capillaries is relatively quick. So you have a couple of seconds, but I think that's where things matters most. That's where things happen when ultrasound hits them.
Well, Sasha, it's interesting that you say that. I mean, at a recent conference we had in the cardiology space, we had quite a bit of discussion about what the downstream effects are from bubble activation, for instance, in resolving microvascular occlusion. And there's quite a bit of interest in this in the sense that even activating bubbles in the bulk of the blood, where you've got large volumes of interactions going on, then stimulate downstream effects. So I think it is important to look at both types of scales, whether you're in the capillaries or you're upstream of the capillaries, and what's the biology that you're influencing as you're moving up and down those scales. So I think you're right. I think we do have to look at it at a variety of different scales.
So perhaps also let me chime in from kind of like a more mechanobiology type of perspective as well. And nowadays, there are actually these microvascular models that are bioactive. And they are indeed perhaps potentially good models for us to try to adopt and try to apply in investigating more about the biological response, perhaps not at a single cell level, but at a capillary level. And I mean, these will actually lead us to new insights as to what is really going on. Going back to Christy's point, like right now, it is true, the biology side of things is still kind of like a black box. Why? Because most of us are actually being trained as a physicist or a chemist, right? But very few of us actually have that expertise in the biology side of things, like mechanobiology, for instance. So it would be very nice if our field actually has someone who is dedicated to the mechanobiology side of things. That would really be important for us to really push into new grounds in that direction.
I also think we need ambassadors to people with expertise like vascular endothelial cells. You can't really plate those and have them function like they do in vivo. And so I do think that that's an area particularly that we need an ambassador that can kind of speak both languages and move into that. I know Inez Beekers has done some beautiful work in Nico de Jong's lab at Erasmus University, where they've got beautiful pictures from the Brandeis camera, but then you see the cellular response, and yet those are still HUVECs that are single cell and probably not attached the way they would be to a basement membrane in vivo. So it would be nice to see some work in that area.
Yeah, so I think one of the questions that got turfed back during the presentations was this question of how do we pull our commercial agent colleagues that are in the diagnostic space along with us and into the therapeutic space. So I want to make sure that we circle back to that question and get thoughts from the group in terms of that, because I think it is in some ways the low-hanging fruit. It's got a little bit lower bar to reaching the therapeutic stage just because of some of the associated regulatory barriers that were discussed. So thoughts related to that?
I think we should let Mike Canny maybe start in, because I think he probably knows this the best.
Is this how to rope in companies that make agents, you're saying, Brian, you're trying to get at? A little bit of that question?
Yeah, it was a question from earlier in Q&A.
Yeah, I mean, I think the hesitancy from a lot of the companies was normal. They have a pretty big market with these agents. There's only a couple on the market, and they didn't want to have any adverse safety effects pop up that could affect their market. So it's pretty normal. But actually, we've been having, as we've been accumulating a lot more human safety data, we've had a really good relationship with several of the companies, actually, and they're really interested in expanding into this space. So I would kind of counteract some of those points and say that they've had a lot of interest, and we haven't had any issues.
Yeah, Eleanor, I think you had your hand up there.
No, it's to absolutely follow that. So early on, one of the things we were told is we don't want you playing with our agent, because if you show that it has a biological effect, we're in trouble. And I think that comes back to something I think is really key, which is showing just how far apart the exposure parameters are and how different the behavior you get in those regimes is. And again, I think this is a very important engagement with our more biological colleagues, where ultrasound is still treated as a single entity. I used ultrasound, and we've got to make sure that we're very clear that if you drive an agent at 1 megahertz at 100 kilopascals, you're going to get a completely different effect to driving it at half a megahertz at 2 megapascals.
And that's a very important point, because I'll say as well, while we're focusing on cavitation nuclei in this session, the two kind of commercial representatives, myself and Mike, we have our own ultrasound device. So that's kind of the one thing that we're not really talking about here, is you can make a fantastic cavitation nuclei, but you need to be able to use it with a device. And that's not going to be a problem if you go with a HIFU device. That's easier. But if you expect to work kind of uniquely with an imaging device, asking them to open up their label claim to be able to utilize that in a therapeutic context, that's not necessarily straightforward or completely answered. So finding a partner that's willing to work with your cavitation nuclei for a particular application isn't a solved problem, I guess I should say. And is one of the reasons why we developed our own device.
Bruce, and Mike, I had a question about that. So in certain types of drug modifications, usually the pharmaceutical companies will incorporate those types of modifications. And I just noticed that with microbubbles in the drug delivery space, there's Oxonics, there's Carthera. These are kind of like very separate entities developing the drug delivery vehicle. Is there, and this might be a silly question, but is there any way that microbubbles could somehow be incorporated as like a modification to a drug that's emerging?
You mean as a new drug?
Essentially, yeah. And it's so, the big difference here would be that there would have to be a major partnership probably with the pharmaceutical agent generator or the company, right? So would that make life easier? I mean, Mike, you're smiling like I'm crazy maybe, but
you're trying to create a lot of work for yourself, I think,
exactly.
is the main answer. There's a lot of aspects there.
So you're saying there's
just a couple
no need to do that.
A couple more things though that we didn't talk about the microbubble agents is the FDA is actually potentially changing the regulatory status of microbubbles and going to classify them as a device. We never talked about that today. I don't know how that's going to affect the field. I don't know if that's going to lower the barrier for new agents or change it. Maybe not, but it's something that's going to happen.
I wish we had Keith Ware here with us today, who's at the FDA, or Shula, who's also at the FDA to participate in that. I didn't even know that.
Yeah, there's a lawsuit that was filed by a certain contrast company, not related to ultrasound contrast, but it went before a judge, and the judge ruled that regulatorily they're driving the FDA toward regulating these contrast agents as a device, which for those of us who've been in the field a while, we're coming full circle. I know certainly Sasha knows this history probably very, very well, but there's a very interesting kind of complication, so to speak. And in some people's minds, it's actually could be advantageous. In other people's minds, it can be a complication. It all depends on your perspective in terms of how to deal with this. And ultimately, we'll have to see. And there's a comment period, I think it may have just closed, going to the FDA about this particular issue. So I think we'll be hearing more about this going forward. Any comments from the panel regarding what's going on in this circumstance?
I can just
historically. Oh, I'm sorry. Go ahead.
Go ahead, Sasha. Okay. Well, I was just going to say that I do think that part of the reason why we haven't seen an agent make it on the market yet is because of just this topic that Brian was discussing. Because your agent and the device, you basically have to go through two. It's a device and a drug. And to most companies, that kind of thought is a nightmare, thinking you'd have to go through two hoops instead of one. So I do think that having a pathway clear to have it be one or the other, just give me one, I think will accelerate the interest, corporate interest, I would think. And then second of all, whoever goes through this gauntlet first is going to be like that big nose on a freight train that pops the cows off the track and then allows a whole bunch of people behind them. Because I think there's so many creative ideas from our community. You just need one to get through, and then it's 510K or whatever the drug equivalent is after that. So that personally is Christie's crystal ball, I think is what's going to happen.
Yeah, I fully agree with Dr. Holland, absolutely correct. And historically, originally, Albunex, the air-filled albumin shell bubble, that was actually approved as a device. And then, because of the lawsuit that Dr. Folks had mentioned, some competitive groups that had lipid bubbles that were considered as quote unquote drugs were unhappy about that, and then hell came loose. The mentioning of the bubbles as whatever and device need to be approved at the same time with that is also an issue. And from the diagnostic imaging standpoint, same thing had happened. When bubbles first came on market, they were not that bad, but imaging side was not ready. Until contrast mode was very good, it was not working very well. So now that you have ultrasound is catching up because you have both equipment and particles ready. So same thing that needs to be orchestrated correctly for what we are doing here now.
Hopefully a lot of this will happen within our lifetime. Paul, please, and then I have a question from the audience I want to pose.
Yeah, I just wanted to quickly emphasize what Dr. Klibanov just said, that for years, the device market has not caught up with the cavitation agents. If we just look at microbubbles for imaging, there hasn't been a big push to focus on microbubble imaging strategies on systems. I mean, many of you guys know if you've got a 20-year-old Siemens Sequoia, that can image bubbles just as well as some of the modern systems. And I think that shows that the industry hasn't really put as much time into developing contrast imaging as they have for other things. And I think that's very much the same for if we're looking at therapeutic applications. It's kind of a chicken and an egg thing. The therapeutic agents haven't been developing, but the therapeutic devices haven't been developing either. So I think moving them both forward is really going to help the field.
I want to pose a question from the audience here to the panel. Will each of these agents, we heard a variety of different agents, nanocups, nanodroplets, bubbles. Will each of these agents require its own specific metrics to dose cavitation or general metrics such as energy? So maybe something that's more generally applicable and ubiquitous, such as energy mentioned by Dr. Coviello, can work across the range of agents. It's an interesting question. I'm not going to wade into that. I will toss that out and let it spin until somebody picks it up.
Go on, I'll start and you can tell me why I'm wrong. I don't think we want to be in a situation where we have one metric for any agent. I think that's going to be a nightmare for everyone. On the other hand, I think, and this has been said a few times, and it was probably the main thing I was trying to get across in the talk, was there are going to be significant differences from application to application, and depending on the level of damage you are intending to do, that is going to dictate the ultrasound exposure parameters and also the monitoring levels, or the indicators in the monitoring that you want to play to. So again, it's not going to be one size fits all for monitoring, but I think we certainly don't want to be in a situation where every single agent has its own regulatory framework. That would be crazy.
I agree with Eleanor, but I think I would go even further. I don't think we know yet what indicator to really use for bubbles and bioeffects. We have cavitation indexes. Everybody kind of defines them differently, but I still haven't seen a good correlation. We haven't compared to thermal dose, for example, nothing close to that. So I think we have lots of basic work we need to still do.
We need to understand what they're doing before we can define a safety measure.
So do you think it's easier or harder in the presence of, let's say, a known agent than if you're relying on endogenous cavitation nuclei?
I would say yes. I think we've got a better chance of predicting what's going to happen. I guess that's going to slightly depend on the agent, though, because the more complex the agent, the more things it could possibly do. And again, not ignoring the biology, then maybe when some of the stuff we've been doing recently, we've come to the conclusion
actually the primary effect is chemical and it's nothing to do with the cavitation. So yeah, I realize that just opens another can of worms, but.
Well, maybe I'll just quickly chime in from a more physics point of view. Right? So, after all, cavitation is a very transient phenomenon, right? I mean, just like turbulence. It's hard for us to come up with a single universal indicator or index to predict what's going on. Okay, say in turbulence, we have Reynolds number, right? We have Schuh's number. But how predictive are they? Okay, so we might have turbulence above 3,000 for Reynolds number, but that's it. The index itself is not very descriptive of the dynamic phenomenon and the transient phenomenon. So it will be very difficult for us to come up with something that is very specific for cavitation.
So I'll take a counter argument to that just for a moment, because this is what makes this all fun, right? Is, look, the mechanical index that we use today is not perfect, but it has utility. So I think in earlier discussions that we had, perfection is the enemy of the good. So we can't look for perfection here, but we have to look for something that will provide us guidance and assistance in this process, right? There's something that we work on together collectively to be able to get us to a point where we have some confidence in its utility and its potential as a feedback mechanism and those kinds of things. So I agree with you, Alfred, and I disagree with you at the same time. I know it's a complex system and I know it's hard, but you got to do something. Christy.
You're very conflicted, I hear.
I want to poke Alfred a little bit too, and that, in order to predict when the onset of turbulence is, we use the Reynolds number. But there's this thing that's a second moment in your field, right? That's the turbulent intensity that correlates quite nicely with turbulence and how much turbulence you get. But at the end of the day, why do we care about turbulence? We care about turbulence because it's going to either reduce drag on a ship or a propeller, right? And so there has to be something practical about why we're using that particular index. I'm just going to go with the turbulence intensity here for a minute. So until we really understand the specific bioeffect, that beneficial bioeffect that we want, we're not going to know what that metric is.
Yeah, but that's quite difficult because there's a range of therapeutic applications. There's a range of different nuclei for those therapeutic applications. So it's still going to be quite complex. But I think if we could ever agree on even flawed for certain applications indices, we're probably not going to make a hell of a lot of progress.
Yeah, that's a common theme. And then Alfred, I'll maybe toss it back to you for.
Yeah, I just want to say, hopefully we can do better than the MI. If we are to ever come up with an index, I think we do have quite a lot of room for
this is a bit of a common theme based on what I have heard from other panel discussions as part of this overall workshop. What is an acceptable sort of index or indice? How is that measured? What's the instrumentation that's utilized for that? How does that relate to the desired bioeffect? I've heard this type of conversation is sort of interwoven amongst all of these panels. And so, if we can ever get there, right, and we all have to come to some level of agreement on what that will be, but then also how does that relate to the desired bioeffects, right? And then before Alfred and Eleanor, is there going to be different indices for different, maybe one or two for specific types of bioeffects of interest or therapies? So that I've heard this conversation before. So Alfred and Eleanor, please.
Yeah, I just want to say maybe, so going along the lines of maybe trying to come up with something better, right? Something related to dose. I think that would be relevant. But then I think what's really missing in all these indices is any incorporation of biophysics or biological knowledge into the entire process. So I think if we are to define something along the lines of those, it would be really nice if we try to bring in some sort of biophysics knowledge here.
And that was the benefit, and then Eleanor, of the mechanical index. It allowed people to sort of unify around something that was very common, right? You can have any laboratory across the world that was working on sort of cavitation, inertial cavitation. They could at least get behind the mechanical index, right, to compare notes. Eleanor, please, and then I saw Kalervo's hand.
Directly written. So the first one was to defend the mechanical index on the grounds, A, it's massively abused by everyone in therapeutic ultrasound who seem to ignore the second half of the paper where it's very clearly set out what you would need to do to modify it for longer pulses and various other things. So I mean, there's, sorry, I don't know why I'm saying this when Christy's on the call, but yeah, please read the second half.
I agree with you.
Yeah.
Well, and I would go on to say before Clairvaux comments that if you look at where the thresholds, especially in the presence of bubbles, where you've got a nuclei distribution and you've got a broad nuclei distribution, then those numbers have hit pretty solidly in terms of where that biological threshold is. And so there, you have to understand the mechanical index in the context in which it was developed, which was in a broad distribution of nuclei. And if you're starved for nuclei, then things get a little bit different. Clairvaux.
Yeah, no, I agree with Brian. I think that the mechanical index works really well predicting for blood-brain barrier modulation, predicting the frequency response, how the bubbles are responding. But then to Alfred's point, I mean, what is the dose? It doesn't predict what the magnitude of the opening or anything like that. So if you have a similar exposure, you can relate the frequencies from one frequency to another and get similar end effect. But it is a complicated. But at least there we have something. So I think that's.
And can I just to link this back to, I think 2 weeks ago, it might have been last week, I'm sorry, I lost track of time, but making sure that there is unification of calibration of equipment for treatment monitoring in everything we do, and actually having real units on some of this would be a huge help, because there is correlation, even in the absence of mechanistic understanding, between the absolute energy emitted by cavitation and bioeffects actually across the board. And that's a good starting place. But yeah, I really want to push for, can we make sure we've got some numbers and some units on our measurements?
Eleanor, I would add that we should make a call to all our colleagues in the field. Every time they write a paper, please report, not just the mechanical index, make sure you're reporting your duty cycle, your pulse length, all those other factors that are not acoustic pressure and frequency, right? That play a substantial role in bioeffects and therapeutic outcomes.
And I know that Gail published a paper in UMB some time ago trying to set out what those should be. I mean, maybe a good outcome for this set of workshops would be to either use that or review that and add to it and just have that as something that we all try and use and encourage everyone to do, because it's a good start.
All right, I got a question from the audience here. Paul expressed an opinion that medical device development for therapeutic application of cavitation was not there yet. What are the needs in terms of medical device then?
Yeah, so that is a great question. So if we're looking towards medical devices that are designed specifically for therapeutics with cavitation agents, what exactly do we need? I don't know that I can answer that question immediately, but certainly we know there are different acoustic parameter ranges that are optimal for therapeutics that are not the same as for imaging. And we saw from Christian at Oxonics earlier, they're building their own system that has imaging capability and a separate array subset for the lower frequency energy for therapeutics. And I think that's an example of the type of improvement that the hardware needs to make as well for us to really optimize therapeutic results with a tool that's not just an off-the-shelf imaging system. I'm hoping someone else can continue that. Christy, I see.
Christy's got her hand up.
Yeah, well, I just have a comment, really. It's about calibrating, receiver arrays for passive cavitation imaging or passive acoustic mapping. My lab has done this actually during COVID. It is not an easy thing to do to try to figure out what the sensitivity of these big arrays is. It's not obvious because oftentimes the connectors, those big boots, in order to make that measurement, you're going to change the impedance by actually connecting it. And it's not obvious how to do that well. And we got into it so much that I'm thinking, gosh, does every lab have to do this in order for us to compare our results? I actually think that that's a long road to hoe. It's not going to be that easy. So if we could come up with a better way to get at those joules, instead of joules, volts squared per megapascal squared, or whatever the other non-calibrated is, I really do think we could probably have an entire conference around what unit it should be and really try to make it easy rather than difficult.
Before Colorbo here, that actually was a topic of discussion at one of the earlier, I think Andrew Morrison, so someone from a company that does metrology, was participating in a conversation, and it was, how do you standardize making the measurement or characterizing you receive array, receive transducers for passive cavitation imaging and mapping. So once again, very common themes are coming up across multiple panels. Colorbo, please.
Yeah, no, I agree with Christy. It's complicated, but doable. But the issue really is not so much the equipment. The issue is the fundamentals there. When you have a bubble cloud, some of the bubbles are oscillating out of phase, some of them are in phase, and you cannot really quantify the effects based on magnitude signal. So it has to be something different. So that's why we are looking in the subharmonic as an internal calibration point for the bubbles, which we are then using as an estimator of the pressure that we use for standardization. But it is just a calibration. It's nothing more for us. But that's a very good point, and we need to understand much better the bubble physics, and especially the clouds, before we can come up with an index.
So I'm going to make a statement here that may or may not be true, but I'd like to know what the panel is thinking about this, which is in medicine very often, for instance, in Doppler, we end up looking at ratios, pulsatility index, resistive index, all of these kinds of things. And so I want to put your hats on just for a moment to think about relative measures rather than absolute measures. Now, I understand the interest in getting to the absolute number and saying how many joules we got delivered at a given location. But when you start talking about needing to understand what the impact is and what types of cavitations are occurring and what those impacts are, is there an opportunity for indices of these ratio types to be able to give you information about a biological endpoint of interest?
I think that's a great idea. In particular, I think it's going to be logarithmic. So I think we're going to lean on, decibel, because at least in the area that I've worked in, which is not I'm working a little bit in histotripsy, but really we're talking about orders of magnitudes of difference of bubbles nucleated over time. So I do think that it will be a ratio, and I think it's going to be logarithmic.
Colorbo, please.
Yeah, I think it's a good idea, and I agree with Chris. It could be logarithmic for histotripsy type things. For like the blood-brain barrier, I think the key is that each location, like if you do multiple sonications like we do, is that you get uniform exposure. So you need to have some way of quantifying that you are having the same exposure So their ratios might matter, and maybe I could indicate.
I saw other nodding heads. Anybody else?
No, I was going to say, I think using some of the actual measures that we have in ultrasound systems to look at the vascular response would be a good thing to do. And actually linking some of those in so that we are trying to measure the biology as well as the cavitation.
What about using as a standard the reaction of a Definity or SonoView or echo contrast agent to a diagnostic pulse, relatively speaking?
Well, if I recall, so with the mechanical index, a lot of it was, built around the physics in terms of the bubble wall dynamics, in terms of velocities and energy. And so would it be possible to link it somehow to relative to different, a particular type of bioeffect, right? So is there a certain amount of cavitation activity that leads to homogenizing tissue, right? Or histotrypsy, which could be on the extreme side compared to something that might be more subtle where, for example, Richard Price has published a couple of papers where he's doing this sonoselective delivery, where you're delivering drugs to the endothelial cells, which is going to be more subtle than that histotrypsy, right? And so you got a dynamic range that you could fit that cavitation activity between, or dose between.
Kirsty, I think that's a good idea. I think that's the best we can do right now, is to use the bubble behavior as an indicator of what our exposure is in vivo and correlate that with the player effects.
So circling back for a moment, because there were some questions out there about dose, but I want to make sure that we don't lose sight that this session is all about exogenous agents and understanding kind of what the impact is. So what types of metrics do you guys think need to be evaluated with regard to these agents to develop kind of a comprehensive profile for a given agent? Pharmacokinetics, level of oscillation of that agent, mechanical properties of the bubbles, or other types of things that you think are the critical metrics for these agents. And I understand that in some cases it will be application specific, but more generally, what do you think should be the commonality of those types of assessments?
First and foremost, safety of those things. So if something goes bad, it will be a significant downside for the field. So we have to be safe. That is the first order of business. So that's what matters most. Again, generally as quote unquote diagnostic imaging dose is considered relatively safe, yet every, 20,000 injections, there will be some undesired side effect even without, even before ultrasound sort of diagnostic imaging happens. So this is, of course, needs to be improved. And of course, bubble shell matters, things like that do matter. So you want to minimize the dose, of course, from the standpoint of contrast agent. You want to minimize the largest bubbles that would clog the capillaries anywhere. Those things matter. You want to minimize undesired complement activation, for instance. So some of those agents are sort of same as liposome nanoparticles that are used for drug delivery in cancer therapy, and they're known to significant amount of those induce complement activation. That's work done by Janusz Zebeny, now in Budapest. And bubbles have much smaller dose than those liposomes used for drug delivery, but still it's an issue. So safety first.
Any other thoughts?
Yeah, anyone else, please?
I think we talked about a lot of the parameters earlier, but we talked about size, circulation persistence, cavitation or activation threshold. And although, for like microbubbles, that might be the same, I'm sure that's very different from the solid type particles, from the phase change particles. And all of those, of course, relate back to safety as well. So, but those are three things that I think would need to be well characterized for any agent, size, pharmacokinetics, and cavitation threshold.
Can I add size as a function of time as well? Because I think the fact that these things change once they're in the body quite dramatically, even before you hit them with ultrasound, definitely once you do, and we shouldn't ignore that. I think with some of the nanobubble stuff, coalescence is probably responsible for a lot of what we're seeing.
Well, I know in one of the slides that we saw today, we definitely saw a persistence in circulation for the nanoscale agent, right, in comparison to the other. And the point was brought up, I think it was Clairvaux's presentation, that maybe there is some other mechanism of retention or something that's keeping them in circulation for an extended period of time. There's also the question of whether or not the normal mechanisms that would be taking them out of circulation, maybe they're a bit stealthier to that in that case. So we have about.
I think this.
Go ahead. Sorry.
So I think that a comment from earlier is kind of putting a lot of this into context. A little bit of a long preamble, but I think it was Mike that said, we need to understand whether these parameters are even relevant for, specific applications and so on. But I think in terms of mechanical dose, I think Dr. Holland and Dr. Yu put it well, which is just, what is the bioeffect that's happening, right? So that's kind of the gauge of, that's the outcome that we want to achieve. But, coming from a biophysics background, somebody else earlier mentioned that, I think it was Dr. Klebanoff, the bubbles are reacting with very complex tissues. Sometimes they're literally squeezed into a capillary, et cetera. And I think that despite having a biophysics background, I think modeling can be really difficult. So if we take a step back from the bioeffect, we have microbubble response, and how microbubble response might affect tissue. And for that, we have things like ultra-harmonic response, monitoring that, Dr. Ninen's group, O'Reilly. And then stepping back from that, we have some of these parameters that, are sometimes not characterized, but they could be characterized to examine or tune microbubble response. And then the way that I see it is at the very beginning, there's kind of the design of parameters or which parameters we want to either collapse or control so that we either need to characterize or don't need to characterize some of these parameters. And I think it kind of flows from there through all the things, all the way to what Dr. Holland mentioned, which is bioeffect. Fortunately, there isn't, as far as I know, a very good way of measuring, a multitude of bioeffects that could be associated with microbubble response in real time. But for sure, I think that we're starting to get there with things like MRI and focused ultrasound, specific types of imaging stuff. Is that too long?
No. To be honest, I think Brian was just alluding to this. We just have only maybe 1 or 2 more minutes. We might have time for just one last question. And actually, I want to pose. There was one question from the question and answer. I think, if I recall, this was after Eleanor, Dr. Stride's presentation, and I think maybe we'll have some closing comments from myself and Brian. Could the stochasticity of nanodroplet vaporization be leveraged for specific applications where some properties like circulation matter more?
I'll take that as I think I put it up. Yes, and actually, we are trying to do that at the moment with an imaging application. Whether it's going to work or not is a different question. But actually, the fact that we know we have a distribution of vaporization thresholds, whatever you want, is quite useful because, you know, you'll get different ones on each pass. I think the big question is, will we reach a point where we have to go above a pressure where the bioeffects are not tolerable? But yeah, I mean, similarly, I think the point's already been made about sometimes having a distribution of bubble sizes is a good thing if you know you've got a distribution of frequencies in your pulse, because you can actually optimize that. So yeah, I think in principle, with the caveat, we need to make sure that we stay safe.
I'll add that, you know, there's various groups that have shown benefit to that stochastic activation of phase change agents, both in super resolution imaging, where you need to look at individual events happening, and also in photoacoustic imaging as well, where some of these are even recondensable, where they'll activate the imaging agent, get that signal, and then that imaging agent will recondense, and they'll be able to do that over and over again. So I think, you know, there are some unique benefits to those kind of agents as well.
That's a very good point, Paul. Thank you for adding to that. So I'm looking back through the question and answer portal. I think a lot of the questions we didn't ask specifically. I just want to point this out, hopefully to the audience. But I think there were a lot of what has been posed or put into the Q&A were part of our overall larger conversation around metrics, around indices, around device development, regulatory hurdles and challenges. And so hopefully a dose, another important issue can be standardization of dose parameter was actually mentioned in the Q&A. So hopefully we've captured all of that in our panel conversation. I think it's been very lively and enriching. I want to toss it to Brian, and then I've actually taken some notes around some of the topics or themes that have really kind of come out from our panel discussion. So Brian, any closing remarks from you?
So I just want to say thank you on behalf of all the organizers for the incredible talks that you guys gave today, along with the discussions. This is the part of this that makes it very useful. We know that going forward, we're going to be looking at trying to compile this information and be able to create some common themes and common understandings, and also be able to point out the challenges that we face and try and do something useful in this field in order to help move it forward, because we know that this is incredibly important. At this point, I'll pitch it over to Fred Padilla, and he can let folks know what's going to be coming up in the next week.
Actually, before you do that, I did want to summarize some things before we go to Fred. This will only take me a minute or two that really fell out of this conversation, as well as from the Q&A.
Okay, go ahead.
And that is, an index, maybe similar to the mechanical index that's agreed upon that people can kind of rally behind and utilize or leverage for communicating results, designing experiments, and comparing bioeffects, right, for very different systems, very different agents. I did hear that from the panel. And then how that index is defined, right, how that index is actually going to be defined. Brian, you mentioned something that might be relative, maybe a relative index. There was a lot of conversation around that. So a lot of head nodding to that idea. And then continued development on the agents and the ultrasound devices, not just the agents, but also the devices and instrumentation side for therapeutic applications. And so those were some of the sort of major storylines that I heard come out of this. I'm gonna make maybe a selfish pitch here just a little bit for maybe representing all of us collectively. I remember some years ago, maybe when Christy was sort of just starting out at University of Cincinnati, there was a lot of work on, cavitation and sort of the deleterious effects maybe in the lungs. And there was a community that was kind of working on that, right, within the AIUM. And then there was a community that was also working on the backscatter coefficient. And there was like these phantoms that were just kind of sent all over the country and around the world. People with different systems were, making these kinds of measurements to try to define the backscatter coefficient. So, it'd be nice if we had that with regards to this index, right? And how that index is defined how that relates to different types of bioeffects, and different groups are working collectively and cooperatively to make that actually a reality. I think that would be extremely powerful. And then there's a technical report, right, that it is ultimately then distributed and posted for the community. So I'd be really excited about, that type of effort, possibly being led by Kullervo Henninen, because he's got all of the tools and all of the toys, and I need to just do a sabbatical and just go basically sit in Kullervo's laboratory for about a year, just soak it all up. So that's the selfish pitch. I'm going to toss it over to Fred for closing remarks.
Thanks, Tyrone. Thanks, Brian. Thank you to all the speakers, panelists of today. It was a great session. The recording of this session will be available early next week. And of course, I would like to encourage you to attend next week's session. It will be focused on the clinical and regulatory experiences, how safety, efficacy are and can be assessed in clinical practice for treatment based on cavitation, namely histotrypsy, blood barrier opening, and drug delivery. And we will also address questions about regulatory approval with people from FDA and other experts in regulatory aspects of therapeutic ultrasound. The session will be moderated by Dr. Alexandra Golby, who's a professor of neurosurgery at Harvard Medical School and who's been involved in BBB opening clinical trials with Graham Woodworth, and by Keith Ware, a research physicist at the FDA in the Office of Science of Engineering Laboratories, and who specialized in therapeutic ultrasound program. So hope to see you next week, same time, same place. Thank you.